We are demonstrating our project entitled the Distributed Laboratory, or subtitled Telemicroscopy. And what we are doing is we are controlling an electron microscope at the UC San Diego School of Medicine from the Siggraph showcase floor. We're using high-speed networking, computer graphics, and supercomputing to acquire images from the EM and to generate stereo, pairs, and tomograms, volume-rendered 3D reconstructions of subcellular structures. The structure that we'll be looking at today is the Golgi apparatus. It's the organelle within the cell that is responsible for adding sugars to newly manufactured proteins, thereby labeling them for delivery to the proper destination within the cell. Well, the two applications that we're going to be demonstrating for you are called Grid Browser and Survey. And we'll get started with Grid Browser. And what Grid Browser is essentially is a database, a graphical database, that enables us to collect numerous images from the electron microscope and catalog them in a hierarchical fashion. And we have lots of graphical cues in the database to tell us what's going on. One of the features of the graphics database we call markers, and they enable us to record interesting locations on the electron microscopic imagery for future reference. We can also very easily move around from one of these markers to another. Another graphical cue that's integrated into the database are called survey markers. And survey markers are little icons that show us where we have collected additional imagery at various magnifications. You can see that we can also scroll around the image using a number of different techniques. And finally, the third type of graphical marker is a tomography marker, which shows us where we have actually computed 3D reconstructions from the electron microscopic data. All right, now we would like to capture a stereo pair from the microscope in San Diego. Phil will be steering the microscope operator and the microscope itself to center an object of interest in the microscope reticle, which you can see is a yellow box on the display. That inscribes the area that will be captured at the current magnification, and we will center an object of interest in the reticle, and with the assistance of the operator in San Diego, we will capture a stereo pair and have it transmitted back here to the show floor. We will use the voice teleconferencing tool to communicate with the microscope operator and isolate the object of interest, and then using a link command in the software, we can update both displays to show exactly what it is that we're going to be capturing.
Thank you. San Diego, are you ready? I'm sorry, San Diego.
Your specimen is up, and we're showing you a low magnification image right now.
Okay, could you link that to me, please?
Okay.
Okay. Do you have a recommended magnification? I'm set at 4KX.
I would suggest 5K.
Okay, I'll change my magnification.
Roger.
It looks good here. Can I go ahead and transmit the request?
Go ahead.
Request transmitted.
All right.
The bulk of the procedure for collecting this stereo image is automated. However, there are a couple of steps that require the operator to intervene, one of which is to focus the microscope. Ultimately, we would love to be able to do things like focus the microscope, align the beam of the microscope, correct astigmatism in the beam, and other sorts of operations that require a high degree of expertise on the part of the operator. We haven't reached that point yet. So currently, when it becomes time to focus or do those other types of operations, a requester appears on the workstation in San Diego asking the operator to please focus, and then click on a little button to tell us that they've finished focusing. At that point, the automatic acquisition of the stereo pair can proceed. The first image of the stereo pair has already been transmitted back to us, and the second one is incoming right now. After that, there's a short period of processing to integrate the two images into a red-blue stereo pair. And if you watch momentarily, we'll see that stereo image appear in the window.
Where's it going to arrive in that?
Right
That's
there.
it.
Well, after you look at that for a minute, why don't you tell us what you think you ought to do next?
The next thing we'll shoot will be a montage that we request.
Okay.
Next, we would like to show you how we can perform a survey of a wide area on the sample. And what Phil is going to do is he is going to direct the software to a particular magnification and a particular geometry of tiles. And then the microscope will automatically collect multiple series of images that will be stitched together into a montage. This is done by deflecting the beam of the microscope, not by moving the specimen on the stage. And the electron optics in the microscope are so accurate that by using that technique, we get a mosaic that perfectly matches up on all sides. Phil?
Okay, San Diego, I've adjusted my reticle for 3x3 with no corners at, I'm still at 5K. Let me link this position to you. Do you have the new position, San Diego?
Yes, we do.
Okay, I'm going to transmit the request.
Okay, now this operation has been initiated. The operator of the microscope in San Diego will only need to focus once, and then the microscope will automatically collect the 5 images that Phil has requested.
So it's a
Those will be
4-second
transmitted
exposure,
back to us
then a 14-second
in a non-blocking fashion so that we can begin to look at the images as soon as they begin to arrive. Okay, the indicator on the screen is showing us that 2 of the 5 images that we have requested have already come in. The red squares represent missing data. The blue squares represent data that has been transmitted. And you can see approximately every 30 seconds or so another image will come in across the network.
Looks like approximately...
When we're normally operating back in San Diego on our FDDI ring, it takes between 1 and 2 seconds for each image to come in. The T3 network is slowing that down to about 30 seconds per image.
I would probably have to take some additional test images to get the that flop out of the alignment.
Yeah, one should pop up in just a moment. There.
Okay, we just received the 4th tile, and it looks very good.
You can get tight on it, and there's one more. You get one more chance.
There it is. It arrived. Looks very good.
Okay, I think you were hearing before I am this end.
Yeah, it gets sent to us first.
Confirm that the image has been sent.
Yes, all 5 images have arrived.
So far we've showed you how we can remotely capture images off the microscope and perform wide area surveys of the sample. Another important feature of this application is the ability to do 3-dimensional reconstructions. We perform the 3D reconstructions of subcellular structures by using tomography. The specimen is on the microscope stage and can be tilted at plus or minus 60 degrees. We can then take that series of images at the different tilt intervals, and a software package called Supreme can create a volume of data from the tilt series. That volume can then be volume rendered, producing a 3D animation of the subcellular structure.
There are 2 goals. There's sort of a local environment goal and then a more distributed general goal. The local environment goal is to take best advantage of the high-performance computing capabilities at the Supercomputer Center by linking our national resource for microscopy so that we can do as much as possible to process these very, very large data sets, to learn a lot about them. Our national resource in San Diego is also a resource funded by the NIH which is supposed to provide service to users outside our local geographic community. By developing this software package that will allow remote users to look at specimens they might have sent us in the mail, we can provide them with rapid information at their university in Illinois, in wherever, about their specimen. If it looks like it's an interesting specimen and they really want to come out and do more intensive work on it, then they can buy an airline ticket, fly out with their specimens.